So just reviewing the events of the resting membrane potential and what are things that you need to know to be able to really connect all of the concepts here. So you need to know that the resting membrane potential value is -70. You should also know what this means, right? That the inside of the neuron, when the neuron is at rest, is negatively charged 70 millivolts than in respect to the outside of the cell. So there's more negative charge inside. And then you also need to know how we establish that. So you need to know the permeability for sodium is less than that of potassium, 25 times so 25 times greater permeability for potassium. In other words, more potassium channels. You should also understand that the resting membrane potential is a combination of the forces acting on sodium and the forces acting on potassium. So you should be able to break out those individual forces and know their direction. So sodium wants to get into the cell because both of its forces want to move into the cell, right? Its CDF and its EDF are inward. So it has a strong positive force into the cell. You should also be able to connect that potassium. Potassium has opposite forces, but its stronger force is out of the cell. And so it's going to be weakly moving out of the cell. The sodium potassium pump is also extremely important. You should know the direction that it pumps. So 3 sodium out, 2 potassium in, and that is going to be in the opposite direction. This is an active pump. This is active transport. So it's moving these ions against the gradients, right? It's electrical and chemical gradients in order to establish this steady state. So when we have all of these factors in play, there is no net accumulation of either sodium or potassium on either side of the neuron, and that is how we establish a more negative environment inside and a more positive environment outside. So really just being able to break down the details of how we get to this -70 millivolts, okay? So especially understanding the individual ECDF of both of these ions. Another thing that's important is understanding the neuron, the structure of the neuron, the types of synapses that we spoke about. So you should know the two general types, electrical versus chemical. And the features of electrical we described were that it's fast and it employs gap junctions and it is bidirectional. For the chemical synapse, there were much more details that we focused on. So that was really the main focal point in terms of looking at the synaptic transmission, right? The signal transduction and synaptic transmission. So when we think about the structure of the neuron, the proximal portion or the cell body is where we have the synapses happening, mainly the axodendritic, that's the most common type, but we can also have axosomatic, and then we can have axoaxonic at the distal end. We want to think about the presynaptic neuron being the neuron before, and the postsynaptic neuron being the neuron that's receiving that synaptic signal. We also want to think about the location of the synapse. And so we're going to look at the details that happens here on the slide coming up. But just understanding where on the neuron this is taking place is really important for understanding how graded potentials differ from action potentials. So the synapse is received at the proximal synaptic junction on the soma, and then various types of graded potentials are resulted based upon the neurotransmitter that's released at the synapse. And so it's important to think about the types of graded potentials as well. We're going to review that in detail coming up, but just knowing where on the neuron those are being received is important. So these can be IPSPs or EPSPs, depending on how they change the membrane potential. But these are much smaller membrane potential changes and they're not maintained, right? They sort of dissipate when they move further away from their original site. Then we get to the axon hillock. This is where neural integration takes place. Okay, this is where all of these signals that are being received are added, and we are assessing whether or not we have enough of a change to get to -55. And if we do, then we begin the action potential at the axon pillar. Now, what are the main structures involved in the action potential? This is going to be the voltage-gated sodium channels. So remember, they're found more commonly along the length of the axon, which really makes sense. So the site where the action potential is found is also the site where we have the abundance of the voltage-gated sodium and potassium channels. The action potential will travel down the length of the neuron, and we talk about the mechanism that is seen, especially on myelinated neurons. So it's going to be called saltatory conduction when it jumps in between those spaces that are covered by myelin. So this can be found in myelinated neurons, and then we have normal neurons that are non-myelinated, and then they will just have the action potential going from cell to cell or from location on the membrane to the next. So that is kind of how the action potential moves down the axon. Now, as far as the axon terminal, this becomes important when we look at the synaptic transmission. So the details of how the action potential is converted to chemical energy at the synapse. So the calcium channels are important here, voltage-gated calcium channels that will convert the action potential into the exocytosis of the neurotransmitter that is found at the axon terminal. Okay. We also want to remember that there are leak channels found throughout the neuron. So while we're focusing on some of the voltage-gated channels, we want to think about the leak channels and the sodium-potassium pump that are crucial in establishing the resting membrane potential. We also want to remember the ligand-gated channels. So when the neurotransmitter crosses the synapse, it's going to bind to a ligand-gated channel on the soma of the subsequent neuron, right? The next neuron in series. And that will bring about the graded potentials on the cell body of that subsequent neuron. All right. So that's kind of really zooming out and looking at the big picture details of the synapse, of the presynaptic neuron, postsynaptic neuron, and the different types of channels that are found throughout the neuron. Let's bring all of that together and think about the action potential. So the action potential is where we convert those graded potentials. If they are strong enough to get us to threshold, then they will automatically be converted into a stronger signal, which is the action potential. So we want to think about the types of graded potentials again, IPSPs being negative changes, which make it harder to actually get to threshold. EPSPs are positive changes, which make it easier. So if we do get to -55, so green is going to be the graded potentials that potentially get us closer. And remember, we want to remind ourselves of the summation that can happen here. So temporal summation and spatial summation are just examples of how we can combine many different types of graded potentials, and that could potentially get us closer to threshold. So if we get to -55, because the voltage-gated sodium channels are also being gated at that membrane potential value, they will open up at -55. Sodium will rush into the cell according to its ECDF, and then we will have the upstroke or the depolarization phase of the action potential. Now, because the inactivation gates on sodium are timed, this is not going to go all the way up to +60, which is where it is attempting to go. Instead, it's going to stop at +30, and then it's going to result in the closure of sodium channels and the opening of potassium channels. Remember, those two events are synchronized because of the timed component on the inactivation gates for sodium and for the gates on potassium. So they're both going to result in the repolarization after 1 millisecond of depolarization. Now we're going to have the red area here. So the downward slope in the membrane potential as potassium moves out of the cell, right, out of the neuron. And so the membrane potential is going to attempt to get to potassium's equilibrium potential, which is way down at -94. And because of the closure of potassium channels and because of the sodium potassium pump, we're not going to actually get down, excuse me, we're not going to actually get down. I apologize here, sorry. All right, so we will never actually get down to potassium's equilibrium potential because the membrane is going to go back to rest after hyperpolarization. So hyperpolarization is the overshooting of the resting membrane potential. This is because potassium channels are still closing. So as they are still closing, the membrane potential is lower than -70. And then when they all eventually close and the sodium potassium pump starts reestablishing the resting membrane potential, that is when we get back to -70. All right. So definitely understand how the sodium potassium pump plays a role here as well. So not just in establishing the resting membrane potential, to begin with, but really just correcting the changes of the membrane potential that are seen after the action potential is over. Now let's remind ourselves here of the mechanisms of the voltage gated sodium channel. So how does the channel look like at rest? When it has the two gates on it, so the activation gate and the inactivation gate. Now, when the membrane potential goes from -70 to -55, so from rest to being depolarized, this is where the channel is capable of opening, but the activation gate is still closed. So if we had a strong enough graded potential or, summation of graded potentials, we could open up the channel, but it's currently at -70, and so the activation gate is closed. Now, when we do get to -55, the activation gate opens, the timer on the inactivation gate goes off. So both gates are open, but importantly, the timer on the inactivation gate starts as soon as we get to -55. And so there's only 1 millisecond of depolarization until that inactivation gate closes. So now in the repolarization phase, here we are at +30, and we're now closing the inactivation gates, which stops any further depolarization. And because this is synchronized to the events on potassium channels, we open up those potassium channels after 1 millisecond as well. And so the membrane potential not only stops depolarizing, but now it begins to repolarize with the movement of potassium. Okay. This is also corresponding to the absolute refractory period. So while these inactivation gates are closed, the channel is open here, but it is inactive. It's incapable of being open because the inactivation gate is really kind of that secondary mechanism to secure the channel and prevent any further depolarization. So even if we had a really strong graded potential, we could not fire another action potential, at least not at this segment of the neuron, until the channel goes back, the membrane potential goes back to rest, and the inactivation gate on the channel is now going to return to this state and be open. All right. So that is how the voltage-gated sodium channels are helping to contribute to the action potential, especially their gating mechanisms. Those two events are very important to understand that they are synchronized so that repolarization happens as this timed event where both channels are allowing for that. Any questions so far? So that's kind of reviewing the resting membrane potential, the graded potentials versus action potentials, and then getting into the gating mechanisms of the action potential. Any questions on those concepts? All right. Okay. Let's look here at the synapse. So the synapse is really focusing in on the junction between excuse me, a presynaptic neuron and a postsynaptic neuron. So what is happening between the action potential arriving at the axon terminal of the presynaptic neuron and that signal being converted into a chemical response and then reconverted on the postsynaptic neuron into an electrical response? So what are the steps or the events that take place at the synapse? So we have the action potential triggering the opening of voltage-gated calcium channels. So calcium can move into the axon terminal. Now, calcium is going to depolarize the membrane here, and that is going to result in exocytosis. So what does that mean? This means that the vesicles are going to move closer to the terminal. And so this is the time that it takes that kind of delays. There's a delay between the signal arriving here and then a response happening on the postsynaptic neuron. And the delay is really owed to the time that it takes for the vesicles to move closer to the membrane and then to release their contents by exocytosis. All right. So once those neurotransmitters are released, they can cross the synaptic cleft, they can bind to their corresponding receptor. And in this instance, the receptor can be of two types. We talked about fast receptors that are also acting as channels. So when the ligand binds, and again, just to kind of connect here, this is the same thing as the ligand-gated channel that we talked about being on the dendrites and the soma of the postsynaptic neuron. So the ligand binds, and depending on what type of receptor it is, if it's a fast receptor, it's going to be a channel. So binding is also going to open up that passageway, and we can have depolarization because of the movement of ions. And then we can automatically have a fast response, which is generating that graded potential. If it's a slow response, we're still going to have the movement of ions, but that's going to take a while because we're going to have a few other players that are sort of in the way. So we have the G protein. It's going to be coupled to a G protein. Then it's going to result in the alpha subunit of that G protein moving off. And then it's going to result in the amplifier enzyme creating a second messenger or some other series of events that then will result in the opening up of a channel. So the receptor is not the channel, and there are several steps in between, and we refer to that as being a metabotropic receptor. So two types of responses can be seen depending on the type of receptor that our ligand or neurotransmitter eventually binds to. Now, after that binding takes place and we have the cellular response, we need to clear the junction of this excess neurotransmitter. So we mentioned three different mechanisms to do that. We have the acetylcholinesterase enzyme, which can degrade the extra neurotransmitter. We have the reuptake molecule, which can recycle that extra neurotransmitter, repackage it into new vesicles. And we have the neurotransmitter simply diffusing away from the junction. So that would be the third option for clearing the synaptic cleft. Let's see. All right, excellent question here. So what are the ions that move through the ligand-gated channel, and why is it different than the electrical synapse? Excellent question. So for the electrical synapse, and I'm actually going to go back to the corresponding slide here. So the electrical synapse is different because it is basically cell to cell. So here's what an electrical synapse looks like. It's one cell, one neuron pressed against the other, and they are connected physically by a gap junction, which is really spanning the membrane of both of these neurons. So the signal here just simply moves through the gap junction, and it doesn't need to be converted into a chemical signal. There is no neurotransmitter involved. It's simply action potential moving through this passageway from cell to cell, right? Ions that are helping with the depolarization moving from cell to cell. So this is why it can also be bidirectional. It can go from one cell and back and forth to the other. And it is also faster, right? It's not relying on the docking of vesicles, the exocytosis, the crossing of the cleft. All of those steps are eliminated because we're not converting it into chemical. It's staying as electrical signals, and it is going from cell to cell much faster and bidirectional. For the chemical synapse, things are much more complex, right? Now we have to convert that action potential into a chemical response by way of releasing the neurotransmitter at the synaptic cleft, and then that has to leave the vesicle, release into the cleft, and then cross the cleft and then bind to its receptor. So that's going to be a different type of response. And now we're going to have the binding of that neurotransmitter to the receptor, helping to bring about the response on the adjacent cell. Okay, so it takes much more time, and it is also transferring the type of energy or signal that's being transmitted. Now let's kind of remind ourselves of the fast versus slow response. So the fast response is the ionotropic receptor, where the receptor is also a channel. So when that neurotransmitter crosses the cleft, binds to this receptor, because it is also a channel, it's going to open up. And usually this is going to result in the movement of sodium and potassium. So sodium is going to be moving into the cell, potassium is going to be moving out of the cell, and more sodium is moving in, which is what helps to result in the depolarization. And so that's a net positive charge entering the cell. And so that is how we can quickly respond with a graded potential. So this is your fast channel or your ionotropic receptor. Now let's compare that to the slow channels, or the slow receptors. So these are called metabolotropic receptors. And these are going to be coupled to a protein, which is kind of like a messenger, right? It's going to have these different subunits. One of those will float off, bind to an enzyme in the membrane. That's going to activate the enzyme. And the enzyme is going to make a second the enzyme is going to make a second messenger, and the second messengers are going to be responsible for turning on or opening up the channel. So now we will have the movement of ions, and then we will have a graded potential. So this case, the channel is different from the receptor. So there are steps in the way, which is what makes this a much slower type of response, right? So just to kind of break out how the electrical signals differ from chemical, and more specifically, how the two types of receptors can bring about responses in terms of the electrical energy being converted into chemical energy that we converted by binding to these receptors. Okay. Any